Yes, so we think that CRISPR enzymes have tremendous therapeutic potential, and this is because they have the possibility of treating genetic diseases. And genetic diseases is a class of diseases that affects millions, hundreds of millions of people, which in general are not amenable to treatments with small molecules. So there's a tremendous unmet medical need here. And CRISPR Therapeutics really have the potential to treat these diseases by correcting gene mutations, inactivating disease-causing genes. So just tremendous therapeutic potential here. And however, before that therapeutic potential can be realized, the delivery problem has to be solved. And so there are many people trying to deliver CRISPR enzymes or CRISPR as DNA or viruses. Our focus has been primarily based on delivering the Cas9 protein, and so we're developing nanoparticle systems, nanoparticle or protein conjugates that can deliver CRISPR enzymes into cells, and we've had some success in mouse models with delivering CRISPR enzymes in the brain and in the muscle tissue. And this actually resulted in a company being spun out called GenEdit that was formed by one of my graduate students, Kunwu Lee, who took some of the technologies, based on some of our early muscle work, he formed GenEdit, which is now doing fairly well.
Awesome. And where do you see the future of CRISPR therapeutics heading, especially because you mentioned it can have such an impact on genetic diseases?
So I think so there's a tremendous amount of activity in the space, and this field is just moving at an incredibly fast rate. So I mean, I think there's several classes of diseases that could potentially be treated with CRISPR therapeutics. The easiest ones are going to be diseases where you just have to knock out a gene. So for example, some forms of atherosclerosis are caused by the overproduction of PCSK9. And so if you can knock out PCSK9, then you have a treatment for atherosclerosis. And then there are other diseases, like there are certain eye diseases where if you knock out a gene, you can have a therapeutic effect. But that's a relatively small class of diseases, it turns out. The major genetic diseases are things like sickle cell and things like that, where you probably need to either correct a gene mutation, but it turns out in the case of sickle cell, you can also knock out a gene and have a therapeutic effect. So that would also be a case where you could knock out a gene and have a therapeutic effect. But in general, the real excitement will be when you can, or the real applications, or the applications will be dramatically enhanced if you can actually correct gene mutations, and things like base editing and things like that have the potential to do that. So I think right now it's a really exciting time because I mean everything is basically on the table because there's been such advances in improving gene editing enzymes, going from things like CRISPR therapy, from the first generation CRISPR, which all it did was just cut DNA, which itself was a big advance. But now we have base editing and prime editing, which can potentially repair disease-causing mutations very efficiently in non-dividing cells. And so then it's just like our imagination is basically the limiting factor. I mean, if you can then deliver these into cells, how efficient that delivery is basically determines the potential therapeutic options. And so I'm very optimistic that I think initially it will be, the treatments will focus around things like orphan genetic diseases, orphan diseases where patient populations are small, which I think itself will be a big advance. But I think in 10 to 20 years it could be addressing large problems that affect human society, like cancer, Alzheimer's, and things like that. So I'm very optimistic about the future of gene editing therapeutics.
So you mentioned delivery systems are definitely a challenge, and as well as the gene targets. How to target the genes without compromising any other genetic information. So in addition to that, what other roadblocks do you see in the CRISPR therapeutics world?
Yeah, it's a great question. So outside of delivery, I would say, but delivery is a huge problem. So I don't want to underestimate or downplay how big that challenge is going to be. But if you could deliver it to cells, there is one other complication that may arise. And that is there could be some toxicology associated with doing the gene editing itself. So if you're just modifying the DNA, even if you're not making a double-stranded break, you're still damaging the genome in a certain way, and that could have some unwanted side effects that would probably we won't really understand what those side effects are until after we do the delivery and things like that, until after that those edits have been made in humans. So it's a little bit hard to exactly gauge how important that is. I'm actually optimistic that that's not going to be a big problem. I honestly think that if we could solve the delivery problem, that would immediately open up a whole range of therapeutics, and I don't see any other major problems outside of the delivery problem, actually.
Well, that's great to hear that at least that challenge really creates a focus point for CRISPR Therapeutics to carry out.
Yeah, no, exactly, because and the key thing with all of the other problems that are not related to delivery, the reason I think progress on those areas is going to happen really, really quickly is because all those problems can be solved essentially in cell culture models. So what happens after you make a double-stranded break, or what happens after you induce a mutation in a cell? All of that can be rigorously studied, and the proteins can be very rapidly evolved to sort of overcome these problems. Whereas the problem with what makes delivery such a challenge is that you have to go into a mouse, you have to go into higher animals. It gets very expensive, and it's very time-consuming to do all this. So that's why that's why delivery is still the big problem.
I see. And now, to observe those adverse effects, for example, you mentioned toxicology of potential genome editing and potential long-term effects. Have there been any studies that sort of run longitudinally to see if there are any adverse effects or any subsequent effects of the genome editing?
Yeah, it's a good question. I mean, I don't think so. Definitely in cell culture there have been, and in general, I would say the results look very encouraging. I mean, I think once the edit has been made, there's going to be an initial DNA damage response, but once that goes away, there's really nothing. It's basically done at that point. So that's at least what happens in cell culture, and in mouse models, it's kind of hard to see a lot of the nuances of toxicology are hard to see in mice, but certainly there hasn't been a lot of toxicology seen in no overt systemic toxicology seen from doing gene editing in mice, and even in places like the brain. I think the really big question that comes to my mind when toxicology comes up has to do with in utero editing. I think that's something where nobody quite that's where I think these type of toxicological problems could be much more significant. Yeah.
Okay. And my last question for the day. So you've mentioned a lot that CRISPR Therapeutics really has the potential to help provide treatment for genetic diseases and rare diseases, and such as sickle cell. How strongly do you feel that CRISPR Therapeutics has the potential of curing most human disease in a future where this technology is perfected and we can target and knock out genes in a very effective and safe way?
Yeah, so that's interesting. So if there's a world where the delivery problem has been completely solved and you could deliver this to all tissues, then what particularly excites me is the fact that CRISPR can be repurposed as a transcription factor, and there is a lot of therapeutics that you can do with transcription factors. So if the delivery problem was completely solved, and I guess for things like transcription factors and stuff like that, you also need repeated delivery. So there could be an immunogenicity problem, but if that problem was also taken care of, then it would impact almost every disease, because once you can start using transcription factors as therapeutics, then everything's on the table at that point. So, but even if you just restrict it to gene editing, you would still be able to hit a lot of the major diseases. I mean, I think things like Alzheimer's, I think would be fair game, and certainly sickle cell, cystic fibrosis, and then the big question is would cancer be amenable to sickle cell? I think there would be several cancers where there are gene mutations, and I think CRISPR could certainly play a role there. So for example, breast cancer, there's a BRCA1 mutation that's genetic, that affects a lot of people. All that certainly could be, I think, amenable to treatment with CRISPR.
Well, thank you, Dr. Murthy, for meeting with us and offering your expert perspective on CRISPR Therapeutics. For more expert interviews, stay tuned on our LinkedIn, Facebook, and Twitter.